It's actually a great pleasure for me to announce in person the winner of this year's Avrion Mitchison Prize. F. Mitchison was a zoologist by training, later became an immunologist and cell biologist, and he was the founding director of the Deutsches Rheuma-Forschungszentrum, the German Rheumatism Research Center, which was founded by a group of people and politically made possible by Albrecht Hasinger. But the scientific accents, the first ones, were set by F. Mitchison. So we thought it's a good idea to have a prize for a young scientist awarded in honor of Avrion Mitchison. And we just publicly announced it, actually, that so everybody can apply for that, as long as you are young, an early career scientist. And we then have an external jury which has a look at all the applications and selects the winner. And this year's winner is Denise Beckmann. Denise actually is honored for her work, about which she will talk in a brief while, which has to do with how synovial fibroblasts exert their pathological role in rheumatoid arthritis. She comes today from the University Hospital of the University of Muenster, and then there in particular for the Institute of Musculoskeletal Medicine, directed by Thomas Paap, an old friend of ours and the DRFZ. Denise has a kind of a typical CV for a medical scientist. She did her diploma in biology at the University of Osnabrück in Germany with a thesis on the Wechselwirkung von verschiedenen Unterheiten der Insekten V-ATPases. So that is how actually an insect enzyme is working. She finished her diploma in 2011 and then did her PhD thesis already in Muenster with Thomas Paap, and already at that time on the role of Lasp1 in rheumatoid arthritis, a story that she will detail out in a moment. After her PhD thesis was finished in 2018, she did a postdoctoral training, just continuing, finishing up this work. And then she went for a real postdoc to the University of California at San Diego, to the lab of Nunzio Bottini, working there in the division of rheumatology, allergy, and immunology from 2019 to 2021. And this year she returned then to Muenster and joined the working group of synovial pathologies and rheumatoid arthritis, again at her old institute. It's a great pleasure for me, Denise, to have you as the winner of this year's competition for the Avrion Mitchison Prize. It's very good that it's somebody from the University of Muenster again, actually. And I'm looking forward to your interesting scientific work on how Lasp1 actually controls the connectivity of the synovial fibroblasts. Congratulations, Denise, and please go ahead with your talk.
Thank you so much. So I'm trying to share my screen. Okay, good afternoon, everyone, and welcome to my presentation entitled The Role of Lasp1 in Inflammatory Synovial Joint Destruction. But first of all, I would like to thank Professor Radbruch for this very kind introduction and the committee for giving me this great award. And now I'm looking forward to present you the results of my PhD thesis. As an introduction, a healthy joint shows intact bone ends. What you can see here, it is covered with a thin layer of cartilage and a thin layer of the synovium. The rheumatoid arthritis joint goes under dramatic changes besides the invasive growth and the hyperplastic synovial membrane, cell activation and chronic inflammation, the number of fibroblast, B and T cells, and macrophages is highly increased. Forming the pannus tissue, these cells are invading into the joint space as well as into the cartilage and the bone matrix. This invasion finally results in the progressive destruction of cartilage and the bone. One open key question of the pathogenesis of rheumatoid arthritis is, what is the cellular and molecular basis of the organ specificity? As a specificity, it's known that early cartilage damage leads to decrease or release of matrix products, and along with the activation of the immune cells, fibroblasts are getting stable activated and can then attach to the cartilage surface and undergo a tumor-like transformation. This phenotype is characterized by a repeat growth and the resistance against apoptosis. The expression of adhesion molecules leads on one hand to an increased migration and proliferation rate, and on the other hand to the secretion of MMPs and osteoclast formation, which results in a destruction of the cartilage and the bone. In our study, we were focused on the adhesion molecule Last1, and Last1 is associated with tumorigenesis and showed effects on cell migration, invasion, and proliferation. The ubiquitously expressed adapter protein is localized at focal adhesion sites along lamina podia formation and stress fibers. Last1 is expressed at basal levels in normal healthy tissue and overexpressed in human breast, ovarian, and prostate cancer. The protein is composed of a limb domain, followed by two F-actin binding neolinker cassettes, a linker region with two phosphorylation sites, and a SHV domain. The only known interaction partner of the limb domain are the hemokine receptor 1 to 4. And for example, the hemokine receptor 2 showed an important role during cell migration. Via its SHV domain, Last1 is binding to several proteins such as LPP, cysteine, dynamine, and ZO2. And these proteins are nucleocytoplasmic shuttle proteins. They're important for cellular movement and gene transcription. First, we looked at the Last1 expression in different arthritic mouse models, and we used two different models. The first model is the TNF-engenic mice, and these mice are overexpressing the human TNF-alpha and develop a polyarthritis, which is comparable to the disease in humans. The second arthritic model is the G6pi model, and mice getting induced with arthritis by the depletion of the regulatory T cells and the injection of the recombinant G6pi. And when you look at the tissue staining and also at the immune processing stainings from isolated fibroblast, and on protein level, you can see that the Last1 expression is upregulated under inflammatory conditions. And furthermore, we could also show that Last1 is located at structures of cell matrix and also cell-to-cell contacts. To investigate these in a little bit more detail, we used a 3-dimensional organ culture system, and all different fibroblasts from the different genotypes were resospanned in a matrigel matrix, placed onto a negative coated plate, and these cells were like for 17 days in culture. After these 17 days, the culture was fixed and stained. And in panel A, you can see the results of an H&E staining. And in B, we were color coding the H&E staining and looked for the cellular distribution of the cells. When you're looking at the results of the wild-type fibroblast, these cells are forming a synovial architecture with lining and sublining layers, and this is comparable to healthy tissue in vivo. And when you look at the color-coded panel, you see that most of the cells, also in this increased image, are located in the lining layers. Last knockout fibroblast showed a similar phenotype, but you can see more clustering cells in the middle of this matrix. In contrast, TNF-genic fibroblasts are unable to form a lining and sublining layer. We see a cellular condensation in the middle of the 3D culture, and also here, these red dots are located in the center of the 3D culture. Importantly, the last one knockout, TNF-genic fibroblast, could rescue this phenotype in spite of this inflammatory background. And we see again a synovial structure with lining and sublining layers. And these results are comparable with the results from the wild-type fibroblasts. Next, we were analyzing the effect of Last1 on the migration. And you're using a modified scratch assay. And as expected, TNF-genic fibroblast had a high migration rate in comparison to wild-type fibroblast, and the loss of Last1 led to a reduction in the migration rate. But more importantly, we see a reduction also in the last knockout TNF-genic fibroblast in spite of the inflammatory background in these cells. To test if this migration phenotype is independent from the proliferation in these cells, we used a cyclone assay and looked for the different proliferation. And we could detect no differences in the proliferation rate between TNF-genic fibroblast and Last1 knockout TNF-genic fibroblast. In contrast, wild-type fibroblast showed a reduction in the proliferation rate, but this was also published before, that they have a lower proliferation rate than, for example, the TNF-genic fibroblasts. To determine the functional effects in a little more detail, we use the electric cell subset impedance sensing system, which is also called ESIS, to analyze the cell behavior over the time. And this technique allows to monitor the cells and how they're forming cell-to-cell contacts. By an increase of the resistance, that means that these cells are forming high numbers of cell-to-cell contacts. And by means of the graph, you can see that Last1 knockout TNF-genic fibroblast form up to 20% more cell-to-cell contacts than TNF-genic fibroblast. And you can also see when you look at the graph that these cell-to-cell contacts should be more stable. So we can conclude that under inflammatory conditions, the Last1 deletion leads to a reduction in the migration, and these cells are forming more cell-to-cell contacts. Next, we were analyzing these contacts in the dynamic of these contacts, and we were transfecting TNF-genic fibroblast and also last knockout TNF-genic fibroblast with live at GFP. And you see that TNF-genic fibroblast forming a cell-to-cell contacts by the formation of lamellipodia, and the interaction are characterized by the arrangement of a zipper-like structure. When you look at the results of the Last1 knockout TNF-genic fibroblast, they're forming very large lamellipodia, much bigger than the TNF-genic fibroblasts, and they're unable to form this zipper-like structure. In panel B, we were quantifying these dynamics, and we looked at the extension and reaction behavior of these cells over the time. And we could say, or could show, that last knockout TNF-genic fibroblasts exhibit significantly higher extension rates and lower retraction rates over the time in comparison to the TNF-genic fibroblasts. David Lee and Hans Skinner could demonstrate in 2007 that Cadherin-11 mediates fibroblast cell-to-cell contacts via the formation of the adhesion junction complex. And the adhesion junction complex was characterized in endothelia and epithelia cells and is composed of a transmembrane protein called Cadherin-11, followed by P120 catenin, beta-catenin, and alpha-catenin. The interaction between Cadherin-11 and P120 is for stabilizing these complex to the plasma membrane, and alpha-catenin acts as an interface between the complex and the alpha-actin cytoskeleton. To verify our results from the functional assays, we were now looking at the effects of the Last1 deletion on the adhesion junction protein levels and the cellular distribution. First, we looked at the protein level from Cadherin-11, P120, and beta-catenin. And we looked at the protein level, and we saw overall no differences in the expression between these proteins between TNF-genic fibroblast and Last1 knockout TNF-genic fibroblasts. Next, we were analyzing the cellular distribution, and we were performing immune staining experiments, and we were staining the adhesion junction proteins in green. And you can see the TNF-genic fibroblast in, for example, P120 and beta-catenin. They're forming this very nicely zipper-like structure, and these proteins are also located in cell-to-cell contacts. When you look at the results from the Last1 knockout TNF-genic fibroblast, these cell-to-cell contacts are totally disrupted and don't show this zipper-like structure. We also quantify this distribution via gray values along one cell-to-cell contact from, for example, the beta-catenin staining. And we could demonstrate that the Last1 knockout TNF-genic fibroblasts show a lower variance in the gray values along one contact in comparison to the TNF-genic fibroblasts. Next, we also looked at the arrangement of the beta-catenin cell-to-cell contacts via live cell imaging and transfected our cells with the beta-catenin construct. And again, you can see that the TNF-genic fibroblasts form this very nicely zipper-like structure over the time, but this zipper-like structure is totally disrupted in the Last1 knockout TNF-genic fibroblasts. Next, we wanted to know if Last1 is also part of the adhesion junction complex, and we performed co-IPs. First, we coupled the beta-catenin antibody to magnetic beads and transferred lysates from wild type and Lasp knockout on these complex between the beads and the antibody. In the pull down, we could detect two well-known interaction partners of beta-catenin, Cadherin-11 and P120, and we could also detect Last1. So we can say that beta-catenin and Last1 are in the same structures. Then we performed the reserve experiment, and we coupled the Last1 antibody to magnetic beads and performed again a pull down. We could detect in the pull down catenin 11 and beta-catenin. And we use the beta-actin as a well-known interaction partner of Last1 as a positive control. Next, we analyzed this connection between these proteins via electron microscopy. And the cytoskeleton from wild-type fibroblast were fixed and stabilized with taxol and phalloidin. And then we labeled the Last1 antibody to large gold particles and the beta-catenin antibody to small gold particles. And in the image, you can very nicely see that these both proteins are in the same structure. So we can conclude, we could show for the first time that Last1 is part of the Tian junction machinery via its interaction to beta-catenin. To examine the role of Last1 deletion in vivo, we looked at the clinical score of our different mice at the age of 12 weeks and comparing the results of the TNF-ingenic mice in black with the results of the Last1 knockout TNF-ingenic mice in red. We see only slightly differences in the total body weight, but we see that the grip strength was decreased and the hindpaw swelling was found to be lower in the double mutant mice, indicating a less severe phenotype. Next, we were using micro-CT analysis and could see that the Last1 knockout TNF-ingenic mice show less bone erosion compared to the TNF-ingenic mice. And then we used these hindpaws and performed sections and performed a thulium blue staining to see the cartilage. And we could see that the total area of inflammation was not different between these two different mice, but the length of attachment of the synovial tissue to the cartilage was decreased in last knockout TNF-ingenic mice, and the destained cartilage was also found to be lower in the Last1 knockout TNF-ingenic mice, indicating a less severe cartilage breakdown. So these mice are much healthier. And with this, I would like to thank the whole institute in Muenster, with the head from Thomas Papp, and all my cooperation partners for the help in the last years, and you for your attention.